Okay, folks, let's get started. I won't say happy Monday because I'm getting in a rut. I think of saying happy this or that, so I'll just say greetings. How's everybody doing? I'm probably in a rut saying that too, I suppose. Nothing like being in a rut. Big group today. This is good. Of course, the nice thing about this classroom compared to Milam is it's a smaller classroom, so you get a bigger feel, and I get a closer feel, like the people back there aren't that far away. If I wanted to throw something, I could actually do that. Okay. Well, I have been moving through things. I'm a little bit behind, but I've actually got an extra day built in on this set of lectures before the exam, so I'm not too rushed. And there's not something that we absolutely have to get done anyway, so it's really driven by what we decide to do here. Last time I talked about structural considerations of components of membranes. I talked about glycerophospholipids, I talked about sphingolipids, I talked about cholesterol, and their structures really are essential for what we think of as a lipid bilayer. So those are important considerations. What I want to do today is talk a little bit about some of the things that we find in membranes and give you a little terminology to go with that. And then I will give you a couple of examples of some interesting membrane proteins and some of the functions that they perform. So that's kind of my plan for the day. When we look at membranes, as I said last time, membranes have things in them besides glycerophospholipids, sphingolipids, and cholesterol. They also have an abundance of proteins, and those proteins are important for the functioning of the cell. And it depends on the cell. So one of the things that we see when we isolate the membranes of different types of cells is we see that they can have very different compositions of proteins. This is a gel separation of proteins from the membranes of various cell types. So A here are the proteins found in the membrane of a red blood cell, and you see a variety of those. We look at B, we see the proteins, or predominant protein that we see in the rods cells of our eyes. And we see that has obviously a very different profile than the other ones here. The third one I can't recall off the top of my head, but the important thing is that obviously different cells have different proteins, and those proteins help serve the individual need of the cell. So it's important then to think about what those membrane proteins are. One of the things I will tell you when we isolate membrane proteins is that some membrane proteins can be extraordinarily abundant. If we look at mitochondrial membranes, for example, they probably have the highest concentration of membrane proteins of about any membrane that's around. And we'll be talking about mitochondria soon, so I hope that you'll see at that time how and why that happens. This is a schematic from your book. I'm not real fond of it, and your book uses some terminology that's a little odd. So I'm going to give you some terminology that's not consistent with some of the things that you see in your book relevant to proteins. When we think about proteins in membranes, we can think of them in about 4 different classes. And you actually see 2 classes on here. Okay, now why they didn't put all 4 on here, I can't tell you. But it depicts for us, first of all, that proteins in membranes can be embedded in the membrane. Okay, we see proteins A, B, and C, and we see that in each case, the protein does what I call projecting through both sides of the lipid bilayer. Okay, both sides. So here's going through. There's one half of the lipid bilayer here. Here's the other half here, and it's going through both sides of it. And all 3 of these do that. When we have a protein that projects through both sides of the lipid bilayer, it's called an integral membrane protein. And your book actually uses that word to describe it. Okay? There are some proteins that project through only one side. And we'll see an example of that in a little bit. But it'll only stick into part of the lipid bilayer. Okay? Those proteins are a lot easier to remove from the lipid bilayer than an integral. If you're going to yank this guy out of here, you've got to do a lot more yanking than if only one part is being held or only one layer of the lipid bilayer is holding it. So proteins that project through only one part of the lipid bilayer are called peripheral membrane proteins. Now there's a third category of proteins we find around membranes, and their examples are actually right here. D and E are examples of what I would call associated membrane proteins. They're associated with the lipid bilayer, but they don't actually stick into it. The reason that we see them associated with the lipid bilayer is because in this case we can see that they are interacting with a protein that actually is in the lipid bilayer. But if I wanted to pull this guy off, I could do so relatively easily. All right? So integral, peripheral, and associated are three types. Okay? The fourth type is one that I will call, and this is one I use, a lot of other people don't use this designation, but I call anchored. Okay? And your book actually uses the designation, but they don't call it a specific class of proteins. An anchored membrane protein is a protein that is not itself embedded in the lipid bilayer, but it's covalently attached to something that is. These associations here are hydrogen bonds. Okay? These aren't covalent attachments. But if we have a protein, and the example I will show you is that there are some proteins that actually have a fatty acid sticking off of them. That fatty acid can embed itself in the lipid bilayer so that the protein is covalently attached to the fatty acid that's in the lipid bilayer. It is associated, it is anchored to it, but it is not itself embedded in any way in the lipid bilayer. Does that make sense? And I'll show you examples of those. I wish all four were on this figure, but they're not. Okay, so 4 different types of membrane proteins. Here is an example of what I think is one of the more interesting membrane proteins. It's a protein called Bacterial Rhodopsin. Okay? Now I'm going to tell you several things about this protein. I'm going to tell you a little bit today, and then after I talk about mitochondria, I'm going to talk about a very interesting thing that you could do with this protein relative to mitochondria. This protein has nothing to do with mitochondria in nature. Okay? In nature, this protein is found in the membrane of bacteria that respond to light. So there are bacteria that respond to light, that generate energy from light. Okay? And the way that they do that, at least some of these bacteria, is by using this protein called Bacterial Rhodopsin. Now, looking at this, you can see schematically that this protein is an integral membrane protein. It projects through both layers of the lipid bilayer. And like many proteins, it has, remember the 7 trans domains from last term that we talked about? It has, and I think there's actually 7 domains here. It would actually be classified as a 7TM, although we're not using that designation for bacteria. But it has 7 transmembrane domains, and those domains, as we will see, can have amino acids that will want to associate with these nonpolar fatty acids on the inside. When we're out on the surface out here or up here, we will see more fatty acids that will be interacting with water molecules. So we'll see the more hydrophobics on the outside, the more hydrophilics on the inside. Okay? This protein is very interesting, and I'm going to just give you a little tantalizing hint about what it does. So, in fact, I'll tell you what it does. This protein has in the middle of it a molecule of vitamin A. A molecule of vitamin A. Now, vitamin A we will talk about later in the term, but vitamin A is a molecule that is very light sensitive. Vitamin A has a series of double bonds in a chain, and those double bonds actually isomerize from trans to cis and back to trans in the presence of light. Okay? So they have in the middle of this chamber right here, there's a molecule of vitamin A. Well, what use or what importance is that? Well, having a molecule that changes configuration with response to light means that you actually can have a little mechanical device in the presence of light. Okay? So if we think about it, What we have with vitamin A in here is we have something that can exist as an all-trans or a series of trans bonds, and when light hits it, it becomes cis. Trans, cis, trans, cis, trans, cis. So the longer this thing is in light, the more this guy is doing this, back and forth. And each time it does this, back and forth, it grabs a proton from the inside and kicks it out. Grabs a proton, kicks it out. Grabs a proton, kicks it out. So light provides a mechanical energy, or the light provides the energy, but it's translated into mechanical energy by vitamin A, which uses that mechanical ability to kick protons out of the cell. Okay? And it goes on in the light. It's a very, very cool thing. Now, the significance of that probably won't make a lot of sense to you at this point, but I want you to keep that in mind because later I'm going to tell you something about how you can use that to make a very interesting organism. Okay? Questions about bacterial rhodopsin? Yes, Shannon?
So when the vitamin A is in the cis, is it all of the bonds in cis?
Good question. So the question is, when the cis isomerization happens, do all of them go cis or what? In general, all of them can, but over the short time span, there's one that predominantly does it. It's called position 11. We'll talk about it later in the term. So there's really only one that's doing that, and that's why you get this bend in the middle, kind of like what my elbow is doing here. But if you leave it in light long enough, all of them will infect, so it'll get all curled up if you do that. Okay? Yes? That's a good question. When you remove it from light, will it be in the trans or the cis? It can be stuck in either position. Either position. I did my master's work in biochemistry. The first time I ever worked in a biochem lab working on vitamin A. And because the molecule is light sensitive, we had to do all of our work in the dark. So my real introduction to biochemistry was very much in the dark. Kind of where I keep my students today, I think. So hopefully not. All right. So that's bacterial rhodopsin. Keep that in your head because I will be talking more about that later. Blah, blah. I don't want to talk about that. We talked last term about porin. Porin. Anybody remember the significance of porin last term? It moves water, okay? And it sort of had that inside-out nature of itself flipped. Remember last term we talked about how proteins have their insides tend to be more nonpolar and the outsides tend to be more polar. But porin was flipped because its inside provided a chamber for water to pass through it. And so that orientation of amino acids in porin was different. And in fact, one of the things that we see with membrane proteins in general is they do have odd configurations of amino acids, the reason being that they're embedded in this nonpolar lipid bilayer. So the configuration of amino acids that we'll see in a protein that is dissolved in the cytoplasm, which is an aqueous environment, will be different than the configurations we would expect to see for something that's embedded in a membrane, which is a very nonpolar environment. Okay? Now we can use that information about the polarity of amino acids to look at the amino acid sequence of a protein and predict whether or not this is a protein that would be dissolved in the cytoplasm or would be embedded in a membrane. Okay? And so some of that is seen here. What are highlighted are amino acids that are fairly nonpolar. And you can see these nonpolar amino acids are very abundant in each of these transmembrane domains. You can see there's a whole bunch of them on this particular guy right here. And all the yellow guys are very nonpolar amino acids. Okay? We're not going to go through, we're not going to predict for the most part, not for the most part, we're not going to predict whether or not something is a protein that we would find in a membrane or not. But I think you should know something about the fact that membrane proteins can be identified by their amino acid composition and orientation as being different from cytoplasmic. Okay, well, prostaglandin H2 synthase is an example of a protein that would have been nice to put on that last figure because looking at this, what kind of a protein would you say this is? It's actually a peripheral. Okay? So a part of the protein, this is a part of the protein here. So the difference between a peripheral And an anchored is that a part of the protein itself is actually in one part of the lipid bilayer. And this is a part of the protein right here. So this guy is not anchored. It's actually peripheral membrane protein. Okay? And the hydrophobic side chains you can see right here. They're interacting with the things up here in this part. Because it's peripheral, it's much more easily removed from the membrane if we're trying to isolate it and do various things with it. Now, your book goes through and talks a little bit about what prostaglandins are and about aspirin and the role of that. And I'll just briefly comment on those here. Later in the term, I will tell you a lot more about prostaglandins and the role of aspirin. But suffice it to say that we bring it up now because they're actually showing us an interesting protein found peripherally located in our membranes. And aspirin plays a role, actually affects this enzyme. This is an enzyme, and this enzyme is responsible for making a class of molecules called prostaglandins. So that last protein that I showed you, that prostaglandin H2 synthase, is responsible for making prostaglandins. Prostaglandins are molecules that have many, many functions. One of the functions we see associated with them is pain and inflammation. Okay? Aspirin inhibits that enzyme. So aspirin is a painkiller because it inhibits the formation of molecules that cause pain and inflammation. They do other things as well. But some of the things that they do are cause pain and inflammation. So you take aspirin if you have a headache. You take aspirin if you have joint aches or joint pain or something like that, because at those sites, your head or your joints or whatever, prostaglandins are being produced. Aspirin inhibits their production and therefore reduces the pain. And as I said, I'll say more about that later in the term. Okay. Now, the last thing, these are what your book calls membrane anchors. Okay? So an anchored protein has to have an anchor. And the anchor is typically a long-chain fatty acid that sort of sticks into that lipid bilayer and just doesn't do anything. It just sticks there. So it literally is an anchor, like an anchor is for a boat holding it in its place. These molecules with these long side chains that you see here serve as anchors for proteins. So in this case, we see the protein attached at this end, the polar end, the protein attached through a cysteine residue. In this case over here, we see protein attached through the sugar residues for a rather complicated glycerolphospholipid. Okay? And out here, we see yet another protein linked through a cis. But in each case, the protein's on one end in blue. Okay? So we see the blue there, and we see the green being the portion projecting into the lipid bilayer. So the green part is the anchor itself. And you'll notice that the anchor is not a part of the protein. The protein is simply attached to it. These are not amino acids out here in the green. Okay. Now, we actually can, as I indicated before, predict from amino acid sequence whether or not a protein will be present in a membrane or not. And I'm just going to give you some very general considerations for this. This is a table that plots what's called the Polarity Scale for identifying transmembrane helices. You're not going to memorize this, don't worry about this. But it shows you some things with numbers that you actually learned last term. Okay? What you see here are energies required to move this amino acid out of a lipid bilayer. Well, look at this. Negative 51.7 kilojoules. How much energy does it take to get this guy out of the lipid bilayer? Okay, it gives off energy to get the hell out of there, right? Okay, this is a very favorable reaction, and that's because, look at this guy. It's very positively charged in the middle of that lipid bilayer. It's nonpolar. It doesn't want to be there. It's getting out. We look at the other end of the spectrum. It takes a lot of energy to remove phenylalanine from the lipid bilayer because phenylalanine itself is very nonpolar. We have to use energy to pull it out because it doesn't want to come out of that lipid bilayer. Okay, so as you can imagine, there's a wide variety of ranges of these, and they basically follow the hydrophilicity or the hydrophobicity, whichever way you want to say it, of the individual amino acids. Okay. And there's a depiction, actually a very simple depiction of a protein projecting through the lipid bilayer. There you see one sort of helical transmembrane domain right there. You see an amino terminus, you see a carboxyl terminus, and there's the lipid bilayer. These guys here in yellow are fairly nonpolar amino acids going through that lipid bilayer. And I'm not going to go through that. Okay. Now, one of the things that we see with membranes, and it's kind of hard to appreciate, people for a long time wondered, you look at a cell, oh, question?
Yeah, on that, so membrane or proteins like that go through, are there usually the carboxyl inside and outside?
Good question. Is the carboxyl usually inside or outside? And it can vary. It kind of depends on how the protein itself got synthesized. So there's not a strict rule. Okay? Okay. Now, what was I going to say? Okay. So when we look at membranes, it's a little hard to gauge what's really happening at the molecular level in that membrane. Okay? What does that mean? Well, one of the things we wonder about membranes is how fluid are they? We've already talked about how fluidity is important, right? Because we said that if we went down to what we thought of as a freezing temperature for a membrane, it was hazardous to the cell. That would sort of suggest to us that fluidity is important. And so people have actually measured, using techniques, how fluid a membrane actually is. The idea is to take a cell in a microscope, okay, and have it either itself stained, have the membrane itself stained with a specific color, or to induce a color by hitting it with a laser. Either way, it works. But in this case, they've had it stained with a color, and then they've bleached it with a laser. So they hit this thing with a laser beam, and then they look at how long it takes this bleached color to basically disperse in the cell. And what they find is that membranes are, in fact, very fluid. Okay? In the order of seconds, minutes, this thing disperses very readily, meaning that these molecules that are embedded in the membrane are moving around all the time. Okay? They're moving around all the time. That movement is important, excuse me, as I said, and not surprisingly, because when we freeze cells, then we have some other problems that arise. Okay, so there's the bleach, there's the recovery time, the bleach in this case being a laser beam, and we measure how long it takes that thing to recover, and we get an idea of how fluid the molecule, the membrane actually is. That brings us to what is described as the fluid mosaic model of membranes. And I think this is one of the easiest models that you can have. Okay? It basically says that membranes act like a fluid. All right? That diffusion happens, that membranes are always mixing and dispersing and so forth. And though we may have things that may be big clunkers, like we might think about a big oil tanker on the ocean that doesn't have as much mobility, the water molecules, or in this case, the phosphoglycerides and the sphingolipids, which compared to water molecules around a tanker, are very rapidly moving around. Okay? Well, if this stuff is moving around, we imagine that these can move around a little bit also, and they do move around a little bit, but not as much as the small guys do. And that's really all that the fluid mosaic model says. Okay? Now, there's another component of that, and it's not technically part of the fluid mosaic model, but it's a characteristic of membranes that I want to make you aware of, and that is that we see two layers. When we think of fluidity, does fluidity mean that this is just going back and forth and over there and not coming over here? Or is it coming over here as well? Well, so there's two different types of diffusion that can happen in that scenario. One is flipping across, one is not flipping across. And it turns out that diffusion can happen in the plane of the lipid bilayer very rapidly and very frequently. But movement across the membrane, that is to move this guy from here up to here, occurs much, much, much more slowly. Okay? It's relatively rare that that happens. And it turns out that there are enzymes that actually catalyze the movement of a lipid from one side to the other side. They're called flipases, which kind of sounds like a middle finger sort of thing, doesn't it? Flipases. So a flipase will move this guy and invert it and put it up here, or move this guy up here and put it down here. All right? Now, those flipases probably have an important role. Not completely sure in every case what they do, but we know they have an important role because when we look at the membranes of different types of cells, we see that there is some bias for certain lipids on one side versus the other side. We can actually, believe it or not, isolate the lipids on one side versus the other side and chemically characterize them and discover, for example, that, well, there's a lot more phosphatidyl-ethanolamine on this layer than there is in this layer. Well, if everything were random, we would expect to see equal. And since we know that that movement by itself is very slow, it tells us that flipases have to be establishing what that relative ratio is. Okay? Shannon?
Is the lipids, does it only move lipids from the bottom to the outside, or is there one on the outside?
Both ways. Both ways. So it can go bottom up, top down. Yep. Okay. So, and again, we don't fully understand in every case why there is a bias for one phospholipid or sphingolipid on one side versus the other side, but the cells seem to think it's pretty important, and they put flipases there to actually make those flips. Okay, somebody asked me last time about what happens if you have an unsaturated fatty acid. Why is it different than a saturated fatty acid? And this actually shows not a glycerolphospholipid, but it's actually showing a fat. There's glycerol, and there's 3 fatty acids attached to it. But the same thing happens in a glycerophospholipid. If I take that and I put an unsaturated fatty acid, and remember the unsaturated fatty acid has that cis bond, and that cis bond causes a bend. So this guy doesn't pack as well as the other one did. So that means that this membrane actually takes a much lower temperature to get it to kind of pack down. The other one starts packing and getting very regular very easily at a higher temperature. So it's for this reason that the glycerophospholipids and sphingolipids that have unsaturated fatty acids in them will have a lower melting temperature of their membranes. Okay, and there's the asymmetry I talked about. No, I won't talk about that there. Okay. Membranes are varying from one cell type to another. Bacteria have associated with their membrane a cell wall that gives some support. And that's not Gesundheit. That's not totally surprising because we think about the bacteria is all out there on its own. It's out there in the environment. It doesn't have a lot of other things surrounding it. So it needs something to help give it some structure. And it uses a structure called peptidoglycans, which are a type of lipoprotein. Peptidoglycan bonds that actually form and give a layer of strength to a membrane. Yes, I'll spell that for you. Peptidoglycan. Now, those bonds are important because it's the peptidoglycan bonds that are actually inhibited by penicillin. Penicillin helps inhibit the formation of peptidoglycan bonds and stops bacteria from being able to make their functional membranes. Membranes, of course, are not unique to the outside of cells. In fact, when we look in eukaryotic organisms, we have organelles. Bacteria don't have organelles. But we have things like a nucleus, we have mitochondria, we have endoplasmic reticulum, and these are organelles because in the microscope we can see them as distinct structures, and they're distinct structures that are surrounded by a membrane. Okay? So those membranes of those organelles also contain these same types of compounds that we're talking about here, which include the glycerophospholipids, the sphingolipids, and the proteins that we've been discussing. Okay, and the last thing I want to point out, and again, this shows something about cholesterol, and I'll be talking more about cholesterol later in the term. But I'll point it up here because this figure shows that there's another way to get things into cells. So last time I pointed out the fact that cells have proteins in their membranes that will allow the passage of specific molecules. The example I gave you was that of a glucose transporter. Okay, would allow glucose into the cell. And there are many proteins that will allow very specific molecules into the cell. This mechanism that you see here is another way of getting things into the cell that does not come through a protein. Those other ones, the glucose is passing through the chamber of the protein. Okay? In this case, this is a process called receptor mediated endocytosis. Receptor mediated endocytosis. I'll put that in the highlights. Okay. This mechanism is commonly used as a way for cells to bring in cholesterol from the bloodstream. It's used to bring in other things as well, including things like fat-soluble vitamins, and again, we'll talk about that later. They come in the form of LDLs. You guys probably, if you've ever had physical and you had your blood checked for cholesterol, they look for LDLs, and your doctor probably said, that's the bad cholesterol. Okay? Well, it's the cholesterol that your body needs to get things into cells. It turns out that the way that this is delivered into cells is cholesterol is packaged up in these complexes called LDLs. The complexes bind to specific receptors on the surface of cells, and these receptors internalize that entire LDL. That's the endocytosis part of that. There's the receptor part of it. So receptor-mediated endocytosis brings in LDLs physically into the cell, and then that gets broken down and the various components distributed across the cell as the cell needs things. Okay, and as I said, I will say more about cholesterol and a little bit about this process later in the term as well. Okay, questions about that? Quiet group today? Yes? How are the LDLs made? The LDLs are actually, these are what are called lipoprotein complexes, and lipoprotein complexes are synthesized by your liver as a way of delivering fat-soluble vitamins, and cholesterol to cells. And again, I'll say a little bit more about that at that time, or later in the term. Okay, how about a song? Before we do the next stuff. I don't have a song about this, so I just pulled out a song that was a general song. It actually relates to some stuff we talked about last term that I thought you guys would enjoy. So let's do that. This is to the tune of the old Coca-Cola song. Everybody know the old Coca-Cola song? I'd like to teach the world to sing in perfect harmony, if I only could. Okay, it's called Give Us Real Things. Please sing with me. I'd like to teach my students about some biochemistry to keep them all from drinking Coke and make them trans fat free. There'd be no taking creatine to help them run so fast. And sucralose and NutraSweet would be things of the past. I'd lower fructose levels in the junk that people eat. And salmon farmed and colored up, I'd work hard to defeat. Organic food is good for you, everybody knows. So let's get rid of factory farms and all the GMOs. Give us real things, some for you, some for me. Biochem's good, you see. Give us real things. Okay, a little anti-something there. I'll probably get in trouble over that one. Okay. The food industry will probably now be out to kill me. You know, I'll be on their hit list. Okay. Well, the lecture here really relates to the things that we talked about in the lecture on Friday and also today. The molecules of membranes are important, and we're not going to talk so much about the molecules as much as we are about the proteins of membranes. And we're going to start to get an idea about how these proteins function and how they give cells some amazing things that they do. All right? Now, I've already hopefully planted the idea in your head that proteins are there in the membrane to do a variety of things, but one of the things that we're going to focus on now is the movement of materials from one side to the other side of the cell. Movement of materials. What are materials? Materials could be sugar molecules. Materials could be ions like sodium or potassium or calcium or protons or things like that. Okay? So we're talking about molecules that are being moved across membranes by proteins. That's important to get a handle on. Okay. Well, when we think about the movement of molecules across membranes, we can get some pretty hairy-looking things. What does this try to tell us here? Okay. What this tells us is that if I try to move something across a membrane, I have to keep in mind the concentration of that material on both sides of the membrane. Okay? So if I try to move, let's say, sodium across a membrane, the concentration on sodium on each side of the membrane will be a factor in the energy I will need to move it. All right? Well, that sort of makes sense. Let's imagine, if you will, that I am at the bottom of a large hill, and I have a bunch of water that I want to get up to a reservoir or to a big tank up on the top of the hill. And the way that I have to get that water up there is I'm on a bicycle pump that is pumping that water. Okay? When I start out, I'm only working against the force of gravity, right? There's nothing in the tank. I just pump and water starts going in. But the more water I start pumping into that, the more water I'm working against. Right? The tank starts getting fuller and fuller and fuller. And the longer I pedal to make that go, the fuller it gets, the harder each cycle of the pumping gets. And that's because the concentration of that water above me is a lot higher than it was when I started. Okay? If I stop pumping, what's the water going to want to do? It's going to roll back out. Okay? Well, that's not exactly what happens with ions. It turns out that ions don't completely evacuate. That is, in the case of the water, all of the water will come down. If I have sodium, let's say, at a high concentration outside of the cell, and I have a low concentration inside of the cell, and I have a protein that has a little hole in it that will let sodium move through it, which way is sodium going to move? It's going to move in, right? It's going to go from high to low. Is all of it going to come in? No. When will it stop? It'll stop when it's equal. There's a good equilibrium for you, right? So when the two concentrations are equal, it will stop. All right? Well, what we're going to find with cells is cells have to do some unusual things. One of the things that they have to do is sometimes they have to move an ion from a low concentration to a higher concentration. Okay? Sodium's a real good example. Cells are very much in the business of pumping out sodium and pumping in potassium. They push sodium out against a concentration gradient, and they bring potassium in against a concentration gradient. If I want to do that in a cell, I can't simply rely on the process of diffusion. The first process, diffusion, will equalize everything, right? I can't make one side higher than the other with simply the process of diffusion because they're going to equalize out. If I want to move against a concentration gradient, I have to provide some external source of energy. I have to provide some external source of energy if I'm moving against a concentration gradient. And I'll remind you again, against a concentration gradient means moving it from lower to a higher concentration. Okay. Well, what I've just described to you are two different types or two different considerations in transport of molecules across membranes. Okay? The first type of transport is something we call passive transport. It's passive in that the only driving force is a concentration gradient. At the end, everything will be at the same concentration. For the specific example I gave you, where I had a protein that had a specific hole that would let only sodium through, it's still diffusion driven, but it's selective. It's only letting sodium through. That process is called facilitated diffusion. So passive transport systems rely on diffusion. And if I have a protein that is specific for something, I have facilitated diffusion. Okay? The net result is the same. If I wait over time, facilitated diffusion will still give me that same concentration of the two as will regular diffusion. When you say, well, what's an example of regular diffusion if I have facilitated diffusion? All right? Well, regular diffusion can happen across membranes. I showed you a plot the other day that it was very unlikely, but if I have an infinite amount of time, eventually the concentration across that membrane will equalize without a protein. So diffusion can happen across the membrane, but extraordinarily slowly. Facilitated diffusion makes it a heck of a lot faster and a heck of a lot more specific. Now, what I haven't done is I haven't described to you the situation where I have to work against a concentration gradient. Okay? Working against a concentration gradient requires an external source of energy. Okay? And I'm going to give you a definition for this type of transport. It's called active transport, that I want you to remember, because if you remember the definition, you will find some things a lot less confusing later. The definition for active transport is that active transport occurs when at least one molecule in a process is moved against a concentration gradient. At least one molecule is moved against a concentration gradient. That is what active transport is about. Now, I tell you about that because you'll become confused later. You might say, well, what's the confusion? If I need to have external energy, when I see ATP involved, I know it's active transport. Okay? Well, if ATP is involved, you can pretty well bet it's active transport. But there are other energy sources that will confuse you. And they will look like diffusion, but they won't exactly be diffusion. Okay? So your guideline is, if at least one molecule is moving against a concentration gradient, you have active transport. So what I'm trying to tell you is that active transport has many different energy sources, not just ATP. Some of the ones I'm going to show you at first involve ATP. Okay. All right. Now, this figure schematically shows us a very simple pump. And by the way, I call these things that move molecules across membranes pumps, because they do just that. They help things to move, just like a pump helps things to move from one place to another. Here's a membrane protein. Okay? Here's a molecule on the outside that comes into this chamber, and the entry of this molecule causes, in this case, a slight conformational change, which causes it now to open on the inside, and the little guy comes squirting in. Excuse me. Okay? That is a very simple mechanism by which a molecule can enter through a protein. All right? Again, we see these small shape changes happening in proteins and giving the proteins the functions that they need. The energy input that is here, okay, can come from a variety of sources. I might have, for example, the entry of this guy right here causes ATP to be cleaved, and the cleavage of ATP causes this movement. Okay? There's other things that might happen as well. Okay. All right. Now, we don't need that. What I want to talk about now is a simple type of transporter. Okay? And this type of transporter has a name. It's called a P-type transporter. A P-type. Okay? P-type transporters derive their name from the fact that during the mechanism of action, which you see on the screen, I'll describe in a second. During their mechanism of action, a phosphoaspartate residue is transiently created. So P-type transporters, they're also called P-type ATPases. P-type transporters have a step in their mechanism where a phosphate becomes transiently attached to the protein. That is the protein, the transporter protein. Okay. Well, let's look at the steps in this process. It looks kind of complicated. It's not bad. Here's the transporter. It's embedded in the membrane. Okay? This transporter that I'm getting ready to describe to you is a calcium transporter. It's called a calcium ATPase. And this transporter is very important in the sarcoplasmic reticulum of muscle cells. If you remember from last term, the sarcoplasmic reticulum is where calcium is stored so that it's not accumulating in the cytoplasm. And when muscular contraction happens, that calcium is released to stimulate the contraction. So after the calcium has gotten out, this transporter has got to grab that calcium and put it back away in the sarcoplasmic reticulum so that the cytoplasmic concentration of calcium stays low. So we're seeing now how this transporter moves that calcium into the sarcoplasmic reticulum against a concentration gradient. This is an active transport process. Okay. Well, here's the protein embedded in the membrane of the sarcoplasmic reticulum. We're out here in the cytoplasm where muscular contraction has happened. We have a high concentration of calcium. We're getting ready to move that calcium back in the sarcoplasmic reticulum. Calcium diffuses into this chamber in the protein, as you see here. 2 calcium ions come in. And the entry of the 2 calcium ions stimulates the binding of ATP. ATP binds at the binding site inside of the transporter. Binding of ATP in turn causes a conformational change, which results in the attachment of one of its phosphates to the aspartate residue on the transporter protein. Okay? That attachment of the phosphate allows ADP to leave, and when ADP leaves, the protein changes configuration. Now calcium goes this way. It came in here. It's now moved out here. And the rest of the process is getting back to the original state. Then, after calcium has been released, there's another conformational change that results in the loss of phosphate. That leads us back here to the empty complex, which then grabs more calcium and completes the cycle. Okay? So this process is the way that calcium is being moved. You'll notice that the cleavage of ATP is occurring up here. Okay? And every step in the process that something else happening causes conformational changes that result ultimately in the movement of the calcium ions into the sarcoplasmic reticulum. The calcium are being moved against the concentration gradient into a higher concentration above. Questions about that? You guys realize that every single day I've let you out at least 1 minute early? Do I get a gold star? I did it again. All right. What's that, Julie? All right. Ooh.
Bacterial Rhodopsin.
Yeah.
Sun might serve as a source of your active transport.
Okay, what?
Sun might serve as an energy source of your active transport.
Absolutely. You're getting in the direction I'm going with it. Yep.
I'm walking this way.
Hi. The sea is parting here.